When gardeners start seeds indoors, one of the most common and frustrating problems is the appearance of long, thin, and fragile seedlings. These plants stretch rapidly toward the light, producing weak stems that bend, collapse, or fall over under their own weight. This condition is widely known as leggy seedlings, but in plant science it is more precisely described as etiolated growth. Although it may appear that the plant is growing quickly, this rapid elongation is actually a survival response triggered by the plant's perception of insufficient light. To truly fix the problem, it is important to understand the complex biological mechanisms that control how seedlings interpret light signals and regulate their growth. Every seed contains a dormant plant embryo, along with stored nutrients in the form of starches, proteins, and lipids. When the seed absorbs water during germination, metabolic processes begin activating. Enzymes break down these stored reserves into simpler molecules such as glucose and amino acids. These compounds provide the energy needed for early growth before the seedling becomes fully capable of photosynthesis. The first stage of development occurs underground. The embryonic root, known as the radicle, grows downward to anchor the plant and begin absorbing water. At the same time, the shoot begins growing upward toward the soil surface. At this stage, the plant's growth direction is regulated by two important biological responses: gravitropism and phototropism. Gravitropism allows the root to grow downward in response to gravity, while phototropism allows the shoot to grow toward light sources. Once the shoot reaches the soil surface and detects light, a major developmental transition occurs. The plant switches from scotomorphogenesis, or growth in darkness, to photomorphogenesis, which is growth controlled by light signals. This transition is critical because it determines how the seedling allocates energy between elongation and structural development. Plants possess highly sensitive light-detecting proteins called photoreceptors. These molecules allow plants to measure light intensity, direction, and wavelength. The most important photoreceptors involved in seedling development include phytochromes, cryptochromes, and phototropins. Phytochromes detect red and far red light, which helps plants determine whether they are shaded by other plants. Cryptochromes detect blue light and regulate stem elongation and leaf development. Phototropins help guide directional growth toward light sources. When a seedling receives strong, balanced light, these photoreceptors trigger genetic signals that activate photomorphogenesis. During this process, stem elongation slows, leaves expand, and chloroplasts develop fully inside leaf cells. The plant begins building strong tissues capable of supporting future growth. However, when light intensity is weak or when the light spectrum is imbalanced, these photoreceptors signal that the plant may be shaded by competitors. In response, the plant activates a survival strategy called the shade-avoidance response. The shade-avoidance response is largely controlled by plant hormones known as auxins and gibberellins. Auxins stimulate cell elongation in the stem by loosening cell walls and allowing cells to expand rapidly, which gibberellins further promote stem elongation and accelerate overall growth. Inside the stem tissues, these hormones activate proteins called expansins, which weaken the bonds between cellulose fibers in the cell wall. This loosening allows cells to stretch quickly as water pressure expands them. Although this process allows the seedling to grow taller quickly, it also leads to structural weakness. The cells become elongated but not thickened, meaning the stem lacks the mechanical strength needed to support the plant. At the same time, insufficient light reduces the plant's ability to produce chlorophyll. Chloroplast development becomes incomplete, resulting in pale green or yellowish leaves. Because photosynthesis remains limited, the plant continues relying heavily on stored energy from the seed. As these energy reserves become depleted, the plant becomes increasingly fragile. If light conditions do not improve soon, the seedling may collapse before it becomes capable of sustaining itself through photosynthesis. Temperature can further intensify this problem. Warm temperatures accelerate metabolic activity and hormone production, increasing the rate of cell elongation. When warmth is combined with low light, seedlings grow extremely tall and thin within just a few days. Another often overlooked factor is mechanical stimulation. In natural environments, seedlings are constantly exposed to wind and subtle physical movement. These mechanical forces activate a biological response known as thigmomorphogenesis. During thigmomorphogenesis, plants detect mechanical stress through specialized mechanoreceptors in their cells. These signals stimulate the plant to produce thicker cell walls and increase lignin deposition in supportive tissues. Lignin is a complex organic polymer that strengthens plant cell walls and provides rigidity to stems and vascular tissues. Without sufficient mechanical stimulation, plants produce less lignin, resulting in weaker stems. Indoor seedlings rarely experience these mechanical signals, which contributes to their fragile structure. Correcting leggy seedlings requires addressing each of these environmental factors systematically. The first and most important solution is providing stronger light. Seedlings require significantly more light than most indoor environments naturally provide. Placing seedlings near a window is often insufficient because window glass reduces light intensity and alters the spectrum of sunlight. Using artificial grow lights designed for plant growth can dramatically improve seedling health. These lights provide the wavelengths required for photosynthesis, particularly blue and red light. Blue light is especially important for controlling stem elongation and promoting compact growth. Grow lights should be positioned relatively close to the seedlings, typically between 5 and 10 centimeters above the plant canopy. Maintaining this distance ensures the seedlings receive enough light intensity to suppress excessive elongation. The second important factor is temperature management. Slightly cooler conditions slow down metabolic processes and reduce hormone-driven stem elongation. Most vegetable seedlings grow best between 18 and 22 degrees Celsius. Air circulation can also help strengthen seedlings. A small fan providing gentle airflow introduces mechanical stimulation that mimics natural wind. This encourages thicker stem development and increased lignin production. If seedlings have already become excessively tall, transplanting them deeper into the soil can help stabilize the plant. Some species, particularly tomatoes, have the ability to produce adventitious roots along buried stems. These new roots increase the plant's ability to absorb water and nutrients while anchoring the plant more securely. Healthy root development is essential for rebuilding strong plant structure. Roots absorb water and essential minerals such as calcium, potassium, and magnesium. Calcium plays a particularly important role in strengthening cell walls by forming calcium pectate within plant tissues. As environmental conditions improve, so the plant gradually transitions from emergency elongation growth back to balanced development. Chloroplasts increase in number and begin producing more chlorophyll. Photosynthesis becomes more efficient, generating sugars that fuel both root expansion and stem thickening. Inside the stem, vascular tissues begin strengthening as additional cellulose and lignin are deposited. The plant reallocates resources toward structural stability rather than vertical escape growth. Within 1 or 2 weeks, new leaves begin emerging darker green and more compact. The stem gradually thickens, and the plant's overall architecture becomes more balanced. This recovery process highlights the remarkable adaptability of plants. Even when environmental conditions are initially poor, plants possess the ability to adjust their growth patterns when conditions improve. Leggy seedlings are therefore not simply a mistaken gardening technique. They are a biological message from the plant indicating that light conditions, temperature, or environmental signals are not optimal. By understanding the physiological mechanisms behind this response, gardeners can create growing environments that support strong and resilient plant development. Healthy seedlings are the foundation of productive gardens. When plants begin life with balanced light, proper temperature, mechanical stimulation, and strong root development, they grow into robust plants capable of producing abundant flowers and fruit throughout the growing season.